Welcome to chapter 19. Now we're going to talk about blood vessels. So we've talked about the heart in chapter 18. And then let's draw this in. That's the heart. And we know that we have oxygen-rich blood that's leaving the heart. These are arteries. And this is leading to our systemic circuit, right, to the body. So we have these arteries. They're carrying oxygenated blood. They get here to the capillary bed, and they're going to drop off the oxygen. We're going to pick up CO2. We're going to put it into the veins, and we're going to drive it back to the heart. There's another process here that we'll talk about in chapter 20, and this is your lymphatic system. So there is some fluid that's picked up by your lymphatics and cleaned before we drop it back in the heart. So usually people will call the blood vessels and the lymphatic circulatory. So it's all the fluid that's returning back to the heart. Okay, so what are blood vessels? Basically, they are structures that begin and end at the heart. If you're an artery, the A, you carry blood away from the heart. If you're a vein, you carry blood towards the heart. So if I'm going to the heart, I'm a vein. And if I'm leaving the heart, I'm an artery. Remember, superior vena cava, inferior vena cava, those are veins. They're draining back to the heart. What's our favorite artery? That's our aorta. It's leaving the heart. But remember, there are exceptions. Pulmonary circulation. So let me highlight that again. Pulmonary circulation. If you remember, so blood comes in through the right atrium, through the right ventricle, goes up to the pulmonary trunk. And then if you remember, the pulmonary trunk splits into arteries. They're arteries because they're leading away from the heart, but if you remember, they're blue in color because they are low, have low amounts of oxygen. And then if you remember what goes back to the left atrium, we have our pulmonary veins because it's going back towards the heart. But if you remember, oops, sorry, these are red and they are highly oxygenized because it just came from the lungs. So this is the only exception, pulmonary circulation, to the red-blue rule that we think about. So we usually think of arteries as red and veins as blue, but we do have some exceptions. The correct terminology is artery is away from the heart, vein is towards the heart. So here's our cardiovascular system. We see our heart, and we're showing you the arteries. So they're showing you that the arteries comes out. Remember, this is the aorta, the major artery that comes out that feeds the systemic system. So your artery, your aorta flows into elastic arteries, muscular arteries, and you see all these branches, right? And you see that we go from a really large diameter. And then once we get down here, this is a much smaller diameter. When we get to the capillaries, this is where we're going to exchange gases, but we don't want to have this huge diameter under very high pressure going straight to our tissue. So we're going to narrow. We're going to go from really large and we're going to narrow till we get to really small. And remember, we call those arterioles before we get to the capillary bed. When we leave the capillary, remember, we're going to go venule, and then the venules are all going to merge together, and we have small veins, and then we have large veins. An example of our large veins would be superior vena cava or inferior vena cava. So what's the structure of the blood vessel wall? Well, I've used the word lumen already, but hopefully we know that this is the space where the blood is flowing. So if this is my blood vessel, this is the wall of the blood vessel, and this is the lumen. And what flows in the lumen, hopefully your response is blood. All right, so lumen is just the space or the opening with which blood is flowing. All right, so the walls of the vessels, except for capillaries, which we'll talk about down the road, but basically we're talking all vessels, are going to have layers. So tunicas, you can think of layers. And we have intima, media, externa. So you should be able to kind of make an inference here where we're saying internal, middle, and external, right? External, I'll get it out. All right, so the internal layer, the most internal layer is going to reduce friction between the vessel wall and the blood. So the very first layer on the inside is going to help reduce friction, kind of like we had in the heart with the endothelial tissue. Then we have the middle, and the middle is going to be lots of muscle. And how do I know it's going to be muscle? Because we're doing either vasoconstriction or vasodilation. That's what the middle level does. And then the external layer, this is going to be collagen fibers. They're going to protect, they're going to reinforce, they're going to anchor the blood vessels. And then we have to talk about this term vasovasorum. Okay, here are the tunicas in a figure form so you can see. Here's the tunica intima made up of endothelial cells. So here's our single layer. Remember, this is the same thing as simple squamous epithelium. Good for exchanging gases, carbon dioxide and CO2. Then you have your middle layer and has lots of muscle and elastic fibers. So here we are with this middle layer. This is where we're going to do vasoconstriction and vasodilation. And then we have this outer layer, collagen fibers, acts as an anchor or support. And then inside the tunica externa, we have vasovasorum, or vessels within vessels. So you can see here we have arteries and we have veins that are helping to feed the tissue that's more internal to the artery and the vein. So we got to keep this muscle alive. Remember, the muscle needs oxygen, right? So that it can contract, because it's going to take the oxygen and the glucose and turn it into ATP. So it's got to be able to have its own blood supply. So again, it's the blood that's flowing through the vessels is going to go and feed tissue. So like here, this is the blood flowing through the lumen. It's going to go out into an arteriole and then eventually go to a capillary. How do I feed this muscle? Well, that's what the vasovasorum is for. So in the lumen, we have blood. That blood will be shunted to an arteriole. It'll go through the capillary bed, and then it'll come out a venule, and then into a vein. So the vasovasorum is what's actually feeding the tissue that's inside the vessel. So the first type of artery we're going to talk about is elastic artery, and the one we're the most familiar with is the aorta. And this can withstand pressure fluctuations. So remember, your left ventricle is going to push blood into your aorta, and it's under high pressure. So your aorta has to be able to expand, and then it has to be able to contract. So remember, that's the elastic tissue. So go over here to elastic tissue. I like these tables where it shows you kind of in bar graph form what type of tissue is present. So all of the arteries that we're going to look at, elastic, muscular, and arterioles, they all have endothelial. You can see that this is a lot more elastic. So why do we call it elastic artery? Because it's got more elastic tissue than the others. These have much less. Smooth muscle. So this one has some smooth muscle. This one has the most smooth muscle. And this is why we call it a muscular artery. And then you have arterial. It still has muscle. It just has a little bit less, right? All right, so what do you need to know? You need to know that elastic arteries have lots of elastic tissue. They withstand pressure fluctuations, and the aorta is a good example. So what about a muscular artery? So muscular arteries have more smooth muscle, which we saw here, and they're active in vasoconstriction. These are the arteries that are going to deliver blood to the body organs. All right, the arteriole is the smallest. So you can see here arteriole. It leads to the capillary beds, and it controls the flow into the capillary bed via vasoconstriction and vasodilation. So remember, in order to do these events, you have to have smooth muscle. So do they all have smooth muscle? Yes. Do they all have elastic tissue? Yes. Do they all have endothelial? Yes. It just depends on the amount. So elastic arteries have lots of elastic tissue. Muscular arteries have lots of smooth muscle. And now we have the table that shows you the blood vessel anatomy from the vein side. So here's the capillary, and you can see, this is something good to look at right now. What is the only type of tissue that makes up capillary? You only have endothelial. It is only one layer of simple squamous. That's why it's really good at dropping off oxygen and picking up CO2. All right, then you have a venule. And you can see the venule has endothelial and it has muscle, but it does not have any elastic. And then you can see the vein here has a little bit of elastic, always has endothelial, has lots of muscle here, or I should say more muscle than the other veins, more muscle tissue. If we wanted to look at just capillaries, these are microscopic blood vessels. They allow, in some cases, for only a single red blood cell to squeeze through that lumen. So capillaries allow for the exchange of substances between the blood and the interstitial fluid. What are they going to exchange? Gases, nutrients, waste, hormones. You have this in all tissues except for cartilage. Do we remember that cartilage and epithelial are avascular? So they don't have capillary beds, they don't have blood supply. Also the cornea and the lens of the eye. Both of these don't have a direct blood supply either. All right, so when I say blood and interstitial fluid, I hope that you can kind of draw this out. I know it's coming up in a picture in just a second. I just want to draw it out. So this is my arteriole. This is my capillary bed. And this is my venule. What do I have down here? I'm going to have tissue. And what is in this space? This is interstitial fluid. So in order for the blood's moving, I want to drop off oxygen. It has to go to the interstitial fluid, and then it gets picked up by the tissue, or the individual cells within the tissue. And then if I have CO2 waste, that's going to go to the interstitial, so it goes from tissue to the interstitial fluid, and then it gets picked up by the venule, and your blood continues on. So when you look at this cardiovascular system here, you should see that if we're coming from the heart, this is under high pressure. So that's why we have elastic arteries like the aorta. And then we branch to muscular arteries, then we branch, and what are we doing? As we move down, we're going from large to small. We get to arteriole. Arterioles lead directly to capillary beds. This is where we exchange gases and nutrients. And then the waste gets picked up by the venule, and then we go from small all the way up to large and return the waste and the CO2 back to the heart. This is under low pressure. Before we move on, we have to talk about different types of capillary beds. So we have 3 structurally different capillary beds, and each one is going to allow for the passage of fluids and solutes, but in different concentrations. So if you're a continuous, this is really tight. These fenestrated have a few pores, and these have really large pores. So let's look at the images and kind of walk through. So a continuous capillary, we said, was kind of tight, right? There aren't a lot of pores. These are abundant in skin and muscles. They have tight junctions. They allow the passage of fluids and some small solutes. We also have these in the brain. If you remember the blood-brain barrier that we talked about in 201, right? So the ones in the brain are different. They're unique. They are totally enclosed with tight junctions. There's no communication. These were those gap junctions. But abundant in the skin and muscle. Why do we want to have a tight capillary? Well, we said capillaries are the site that we drop off and pick up. So we drop off nutrients and we pick up waste. Well, remember, your skin is exposed to outside kind of pathogens, right? Bacteria, fungus. So we don't want to be able to pick up every pathogen. We want it to be maybe picked up by a macrophage and maybe digested first. So we don't want to just take anything out of our skin and muscles and put it into a capillary bed. So here you can see a continuous capillary bed. Here are the endothelial cells. And you can see there are little junctions here. There's little gaps, but you can see there's no holes here. There's no holes or pores inside the endothelial cells. The second type we have is called fenestrated capillaries. There are some pores inside the endothelial cells. This just makes the capillaries more permeable than continuous capillaries. These are going to be used if you want to absorb small, let's say small to medium solutes. So things that we might want to pick up from our small intestines, right? Remember, our small intestines are going to have something to do with food absorption. Endocrine glands, maybe we want to pick up a hormone and have it circulating in our bloodstream. Kidneys, we'll talk more about urinary, but there's things that you may want to keep. You may want to keep sodium, you may want to keep potassium, you may want to keep calcium. So fenestrated capillaries is if you want small to medium solutes to be able to be picked up in the capillary bed. You can see here in the fenestrated capillary, you still have the gap junctions, but now you have these pores, right? So you have fenestrations or pores in your endothelial cells. So that's going to increase the size of what you can reabsorb. Now we have sinusoidal capillaries. This is the third type. These are called leaky. So they have fewer tight junctions, really large lumens, and they allow for large molecules, such as blood cells, to pass between the blood and the surrounding tissue. So you find these type of capillaries in the liver. Basically in the liver, we're picking up, I'm simplifying this, we're picking up dirty blood and we're cleaning it in the liver. In the bone marrow, what has to be able to get in and out? Remember, the bone marrow is where we have our hemocytoblasts. And our hemocytoblasts are going to make what? Red blood cells, white blood cells, also platelets. So we have to be able to have an exchange in that bone marrow to get those cells out. The spleen is where we store red blood cells when we break down the components, but we also store some red blood cells. So think larger molecules, blood cells that we want to be able to pass in and out of the capillary bed. Also macrophages. So remember, your macrophages stem from monocytes, and those monocytes are created, they're still white blood cells, right? They're agranulocytes. So these are white blood cells that have to get out. So sinusoidal, think of leaky, really large molecules. So you can see in this picture, we have giant fenestrations, really large gaps, right? So sinusoidal capillaries, most permeable, areas where you need to have large molecule movement. Now we get to move on to capillary beds. So what are capillary beds? They are microcirculation between arterioles and venules. So when they say microcirculation, they're saying small. They're not talking about systemic or pulmonary circulation, right? That's huge. That's large, right? Large scale. So these guys are large scale. This, they're talking about small scale. So flow of blood through a capillary bed, and there are two different types of vessels here, and the picture probably helps a little bit more. There's something called a vascular shunt, where you have a channel that connects the arteriole directly with the venule, and they have something called a meta-arteriole. This is a channel that goes through. Or you can have a true capillary bed where the vessels are all exchanging in the capillary bed. So let's look at the picture. So here, let's do this one down here at the bottom. So this one at the bottom, can you see how the arteriole has only one meta, I call it meta-arteriole, and it goes directly to the venule. So here, these are the sphincters, and the sphincters are closed, and so they're stopping blood from flowing into the entire capillary bed. This up here, we can see that the arteriole, the sphincters are open, and the arteriole is flowing or pushing the blood into the entire capillary bed. So they'll call these true capillary beds, right? Everything's moving through. This one, the blood flows through the middle and it bypasses the true capillaries. So real quick, why would you want to have this difference? Why would you want to have sphincters that are either open or sphincters that are closed? So let's say you're working out and this capillary bed is feeding a muscle. Well, every single point here, you can drop off oxygen. Over here, you can pick up CO2. Drop off, drop off. So you have lots of surface area to drop off oxygen and nutrients. And you have lots of surface area that you can pick up CO2 and waste. This one down here, let's say you are using your muscles, you're running, Do I want to have blood flow going to my organs in this fashion? So if I'm running, what happens to, say, urine output? If you're running, urine output goes down. We're probably shunting off the capillary bed and we're just having blood flow through so the tissue stays alive, but we're not pushing blood out into all of the capillary bed. Next, we have venules. This is when the capillary beds come back together. That's the venule, right? They're very porous. It allows for fluids and white blood cells and lots of items to be picked up into the venule. Okay, so we know that veins carry blood away from the heart. So I'm just going to draw the heart here. We know that we're going away. That's an artery, right? And we know if we're going back to, let's say this is the body. You're going back to, that's a vein. So we know that capillary beds unite to form venules and venules unite to form veins. Do we remember that this artery is under high pressure and the veins are under low pressure? So we have to have adaptations to ensure that the blood makes it back to the heart. This way it's flowing, it's under high pressure. This way it's kind of trickling back in the veins. So we have a few adaptations that we're going to look at. So one, the veins have larger lumens. It offers little resistance. The blood can flow. So blood can flow without, I call resistance, bumps in the road. We have valves that prevent the backflow of blood. So valves are absent in arteries. We only have them in veins. Here's what I mean by larger lumens. So here's the artery, and here's the vein. So even though the vein looks kind of squishy, like it's right, it's not a perfectly round circle, the vein looks a little flabby and flat, it still has a larger lumen compared to the artery. Remember that veins have lower blood pressure than arteries. You can also see that the artery has increased muscle. So the artery has a little bit more muscle than a vein does, because arteries are going to do what? They can do vasoconstriction or they can do vasodilation. Again, we have some clinical correlations. Varicose veins, this is if your valves are not working properly, if they're leaky, incompetent, they call them. There are genetic factors and there's also environmental. So if you stand for too long of a period of time on your feet, that can cause varicose veins. Again, if you're standing, you're putting pressure on your lower extremities. And the blood is trying to push its way back to the heart. So we'll see in a second why that can cause problems. Also hemorrhoids, we'll come back and talk about these when we do digestion. But right now it's the same thing. If you have increased pressure in a vein, that can cause hemorrhoids to swell, and then they can protrude from the anus. All right, now for some fancy anatomy words, anastomoses, right? So basically all we're saying is that this term means interconnections of blood vessels. So basically how blood vessels come together. So what would be an arterial anastomoses? This would be alternate pathways to ensure continuous blood flow. So they're saying if you have an artery and that artery goes to has multiple branches, that's an arterial anastomoses. We have these in the heart. You know how off of the aorta we have the left coronary artery, and off of the right side we have the right coronary, I'm going to abbreviate, artery. So you have two pathways, and both of these pathways are feeding the heart. That's all they're when they say collateral channels. All right, a venous anastomoses. So vein connections or branchings or interconnections. These are numerous. This is so if you have an occluded vein, it rarely blocks up your blood flow entirely. So you're still going to have some blood flowing back to the heart. The third is your arteriovenous, artery, vein, anastomoses. This is a capillary bed. So a capillary bed is where the artery and the vein come together or branch or interconnect. So basically, fancy term for interconnections. Okay, let's look at some terms. We have here blood flow. So I'm going to write BF for blood flow. Blood flowing through a vessel or an organ. So come down here. Varies widely throughout the individual based on needs. So if you are working out, you should have increased blood flow to muscles. Good. If you're also working out, you should have decreased blood flow to the kidneys. Why? Because we want to reduce urine output. If I'm working out, I don't want to have to stop to urinate. So you can change the blood flow depending on the organ or based on the need. Now back to this where it says entire circulation. Entire circulation. That's the same thing as cardiac output. If we're talking about blood flow and we're talking about the whole body, then blood flow is equal to cardiac output. If you remember cardiac output, we measured in liters per minute. It was your blood flowing per minute. Another term that we have to know is blood pressure. So this is the force per unit area exerted on the wall of a blood vessel by the blood. So if I have my blood vessel here, and this is my lumen, and the blood is flowing through the lumen, right? The force as it pushes on the wall, that force pushing on the wall is called blood pressure. So the force exerted on the wall of a blood vessel by the blood. We usually express it in millimeters of mercury. It is measured as systemic arterial blood pressure in the large arteries near the heart. So when we're taking blood pressure, we're usually doing it in the arm. We're usually doing brachial, right? But remember, in the arteries nearest the heart, this is when the blood pressure is the highest. So this blood pressure is the driving force that keeps blood moving from higher to low. So if we're trying to get the blood from the heart all the way down to our toes, our blood pressure has to be high enough for the blood to make it to your toes. Another term we have to talk about is resistance. So you'll either see it as R or you'll see it as peripheral resistance, PR. So this is the amount of friction the blood encounters as it moves through a vessel. I think of this, I just mentioned this, bumps in the road. So the more bumps you encounter, that's going to cause you to go slower. So what are important sources of resistance when we're talking about blood? So one is blood viscosity. If your blood is thick and sticky, your blood is going to encounter more resistance. So it's going to cause increased resistance if your blood is slow. 2, total blood vessel length. The longer the vessel, the greater resistance. So if you have to travel this distance, the chance of you running into bumps is increased. If I only have to go this distance, what's the chance of me running into a bump? It's pretty low. So the longer the vessel, the greater the resistance. And then third, blood vessel diameter. So if we change the diameter, that's going to affect resistance. So in this one, they're talking about fatty plaque buildup. So what they're saying is if you have a blood vessel and all of a sudden I have a bunch of plaque right here, that plaque acts as a bump in the road. That plaque is resistance. It changes the amount of blood that can flow in the lumen. All right, so let's talk about relationship between all of these things. It says blood flow is directly proportional to blood pressure. So they're saying if you have increased blood pressure, that means you're also going to have increased blood flow. And remember, blood flow is the same thing for us as cardiac output. Blood flow is inversely proportional to resistance. So if you have increased resistance, that's going to cause blood flow to decrease. So the more bumps in the road, your blood's going to slow down. So increased bumps, blood slows. And since we know there's a relationship up here, so what happens if your resistance is high? You have lots of bumps in the road. Your blood's going to slow down. If your blood slows down, what do we call that? Decreased blood pressure. So they're all related. So let's do this again. So if you have increased resistance, that means you're going to have decreased blood flow, and that means you're going to have decreased blood pressure. What happens if you have decreased resistance? You have a nice smooth surface. You're going to have increased blood flow, and you're going to have increased blood pressure. This one up here I think is pretty easy. If you have increased blood pressure, you have increased blood flow. If you have decreased blood pressure, you have decreased blood flow. So one thing that I want to tie into this part is, do we understand if we have increased water, that's going to cause increased blood volume, which is going to cause, increased blood flow, which is going to cause increased blood pressure. Can you tie in water to blood pressure? So if I'm reabsorbing more water, I'm going to have increased blood volume, increased blood flow, and that's going to cause my blood pressure to go up. So what's the opposite? If I have decreased water, what happens to my blood volume? It goes down. What happens to my blood flow? It goes down. What happens to my blood pressure? It goes down. This here, this resistance, this is only true if your blood vessels don't change shape. Well, so diameter. So if you do, so I don't know how to word this correctly, that you maybe I'll have to repeat it more than once, but resistance is only the opposite of blood flow only if diameter of vessel doesn't change. I'll show you the picture in just a second. So the more bumps in the road you have, your blood flow is going to go down, your blood pressure is going to go down. If you have a nice smooth surface, resistance is down, your blood flow is going to speed up, and your blood pressure is going to speed up. This only holds true if you don't change the blood vessel shape. So systemic blood pressure. So this is the pumping action as the heart generates blood flow. Remember, systemic pressure is highest at the aorta, and it declines through the pathway. What do I mean by pathway? You start at the aorta, you go through the body, and then what are you trying to do? You're going back through the superior or inferior vena cava back to the heart. So they're saying the pressure is the highest here, and the pressure is the lowest as it returns to the heart. It is 0 degrees, or 0 millimeters of mercury going back to the right atrium. So by the time you dump back into the heart, your blood is under super low pressure or none at all. So I think this is a nice figure. They're showing you blood pressure, and they're showing you systolic. Remember, systolic, you should be thinking contraction, probably associating it with ventricle contraction. And then here's diastolic pressure. You should be associating this with relaxation of the ventricles, right? So look here at the aorta. This is when your pressure is the highest. Let me clear this. So here's your aorta. This is when your pressure is the highest. What happens to your pressure as it goes from artery to arterial to capillary to venule to vein back to vena cava, either superior vena cava or inferior vena cava? We can see that the pressure is almost at 0. So pressure is the highest at the aorta, and it's at the lowest as it returns to the heart. So really just putting into words what we said, When the left ventricle contracts, the blood is forced into the aorta, and we have our peak pressure called our systolic pressure. This is the normal. Systolic pressure, pressure exerted during ventricle contraction. Diastolic pressure, pressure during relaxation. So diastolic pressure, when the blood is prevented from flowing back into the ventricles, and we have the closed semilunar valves, and the aorta recoils. Normal is about 80. It can vary. Okay, here's the same figure again, looking at blood pressure. Here is my systolic pressure. So that's when the ventricles contract. And then here is my diastolic pressure. This is when the ventricles relax. And I'm looking right here at my aorta. So this is the blood that was just ejected. This is the pressure as it was ejected and put into the aorta. And then this is when the aorta relaxes, when it does its recoil, right? And then again, you can see that those pressures will get smaller and smaller, will continue to be lowered until we reach the vena cava. Remember these vena cavas, this is the superior vena cava, inferior vena cava, these are going directly to the heart. We do not want blood coming back to the heart at full speed. We want it trickling back. If not, you can damage the heart. Some more terms here. If we talk about your pulse, what's your pulse pressure? It's the difference between your systolic and diastolic. So your pulse is when you have the throbbing of the arteries due to the difference in the pulse pressure. So remember, 120 pressure comes through the artery, and when that pressure passes, it now goes down to 80 if we're talking about the aorta. So this difference, that's your pulse. That's what you're listening to when you feel the bump, 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 bump. It's from high to low, and that pulse is the throbbing of the arteries as the pressure changes. So capillary pressure. We could go back to the chart and you could look at it. It's about 35 as it enters the capillary bed, and it's about 17 as it leaves the capillary bed. So what we're saying is if we're on the arterial side going into the capillary bed, and then we're coming out on the venual side, on this side the pressure is 35, and on this side the pressure is 17. Low capillary pressure is desirable because high blood pressure would rupture the thin walls of the capillaries. Remember, they're only simple squamous. It's one layer of cells. Most capillaries are very permeable, so the low pressure forces the filtrate into the interstitial space. So what they're saying is we have to get to the interstitial fluid, and then we have to get to the cells in the tissue. So the blood's flowing in the arterial, the blood has to drop to the interstitial fluid, then it has to get into the cells or the tissue. And then remember, we've got our waste that we've got to pick up. It also has to travel through the interstitial fluid and then get back into the capillary bed. So we want to make sure that we are pushing the oxygen and the nutrients into, and that we're picking up the CO2 and the waste, and it is directly involved with blood pressure. So here we're talking about venous blood pressure. So remember, this is low pressure, artery under high pressure. So changes little during the cardiac cycle, small pressure difference, only about 15. Low pressure due to the cumulative effects of resistance. Remember, you're traveling that long tube, right? You're going from the heart to the body, back up to the heart. So this is a really long pathway. And what did we say about length? As you increase the length, you increase resistance. If you increase resistance, that means you decrease blood flow, and that means you decrease blood pressure. So as the blood is coming back to the heart, it's under low pressure. The energy of the blood pressure is lost as heat during each circuit. So some of the kinetics, as the blood is flowing and it's under high pressure, as it gets to each circuit and gets dropped off, you're losing some of that energy. Low pressure on the venous side requires adaptations to help with venous return. So we're going to look at some of these adaptations. Basically, if you're trying to push up against gravity to get back to the heart, you have to have some adaptations. All right, these are some of the factors that aid in venous return. So the first one is probably a valve. So here's our blood flow. Remember, if we're in our lower extremities, we're trying to push up against gravity because we're trying to get back to the heart. But remember, this whole system, once we get to a vein, it's under low pressure. So what are we going to do to make sure that all the blood from our lower legs gets back to the heart? One, we have valves, and these two pumps actually help us. So respiratory pump, when you breathe in, when you're breathing, you are moving blood towards the heart. So take a really deep breath, go. You're actually pulling fluid and moving blood towards the heart. So you're basically pulling blood up in your venous system. And this little valve closes. So I draw blood up, the pressure closes the valve, and now hopefully the blood cannot leak. So hopefully there's no leaking blood. That's when we have faulty valves. That's where you might have spider veins or varicose veins, right? So you push the fluid up, the trapdoor closes, and hopefully nothing comes back down. Same thing with the muscular pump. If you contract your skeletal muscles, they actually push blood up, and then hopefully the valve closes and we don't let blood come back down. So they talk about the skeletal muscles can milk the blood towards the heart and prevent backflow due to the valves. So think about it for a second. If you have a patient who's bedridden, usually you're going to have some sort of physical therapy, and they come in and they're moving the arms and the legs. Or they might instruct the patient to do some point and flex, right? When you do point and flex, you're contracting muscles and you're trying to get the muscles to pump or milk the blood towards the heart so that you don't have any pooling in the extremities. All right, I'm going to stop for now, take a break. We're going to look at maintaining blood pressure in part 2. So in part 2, we'll come and we'll talk about how there's changes in blood volume, cardiac output, peripheral resistance, and how we are going to regulate blood pressure as well.